Hi, I'm Emily Miller. My co-authors are Susan Lisson, Celia Smith, and Kyle Van Houten. And I'm going to talk about using herbaria macroalgae as environmental proxies of historical upwelling in Central California. The study was conducted at and funded by the Monterey Bay Aquarium. So we need real-world observations in order to make models that describe our environment and can predict our future environment. In this figure from the IPCC, the observed records of Arctic sea ice are in purple in the middle, just a small little segment, and the red and blue are two projected scenarios based on that historical, observed, and modeled data. But we are limited by our observed data because instrumentation only began in the latter half of the 20th century. Extending this data further in history would further refine the certainty of our future predictions. Though we are limited by the advent of instruments, living tissues store information about the environment. The width of tree rings record patterns in annual rainfall. This tree cross-section from the University of Arkansas recorded precipitation events that match up with known events in Virginia early colonial history. Similarly, preserved animal tissue can be used to reconstruct entire paleo communities using stable isotope analyses. These are the ideas behind the Monterey Bay Aquarium's Ocean Memory Lab, that we can unlock historical environmental data or memories from living tissues themselves. At the Ocean Memory Lab, we have donated specimens like the gray whale bulla or inner ear bones, such as that in the upper left. The bulla is formed by accreted layers like tree rings. So by analyzing the layers from just one long-lived organism, we can construct a historical time series. We've collaborated with other institutions on seabird feathers collected across years, like the image in the lower left, such that we have each organism providing a separate snapshot in time. And we have specimens that we have collected, like the pressed seaweeds on the right, or macroalgae, that similarly serve as snapshots in time that we can stitch together. Many institutions around the world have herbaria that go back to the 1800s, if not earlier, for some places in Europe. This is the collection from Stanford University's Hopkins Marine Station, the Gilbert M. Smith collection, which is right next door to the Monterey Bay Aquarium. And its collection goes back to 1892, its founding. Herbaria are housed in institutions located across the world. The Victorian era encouraged an interest in natural history, especially in Western Europe and North America. This was a time of curiosity cabinets and time spent studying the natural world, both in the academic world of systematic collecting for documentation, but also for the general public, mostly for the upper and middle class with leisure time. And seaweed collecting was especially acceptable for women. Animals were still in the realm of hunting for natural history collection, which was often restricted to men. Intertidal exploration was accessible for women. You didn't need specialized equipment, and socially acceptable. Margaret Gatty, pictured here, wrote the herbarium collection British Seaweeds, and wrote publicly advising women how to collect seaweed while still following their gender norms, like wearing a corset and having a gentleman chaperone to serve as lookout. These are some of the plates from Gatty's substantial documentation of British seaweeds. And some are very artistic and almost look more like scrapbooks, such as this one, or really unique, like this one that is bound between whale vertebrae. So we wanted to have a consistent time series back through time, and the Monterey Bay Aquarium began in 1984, and so their pressed collection begins then and was based around specific projects. Neighboring Hopkins Marine Station provided the bulk of our data set and had pressings from its founding in 1892 to around 1980, when Professor Isabel Abbott left, who was collecting in large part for her textbook. And then using the online database resource macroalgae.org, I reached out to 4 other institutions: UC Berkeley's University Herbarium, San Diego Natural History Museum, University of North Carolina Chapel Hill's Herbarium, and the University of Michigan's Herbarium, that also had specimens from our region that filled in some of the time gaps that we had throughout our time series. So this study would not have been possible without these herbaria and their curators, who were excited to share this data with us. And we're also grateful to the collectors that made these collections possible. Some of the most beautiful specimens in the Smith collection at Hopkins were collected by Mrs. Swanton, that's all it says, who was either the wife or mother of Fred Swanton, who was mayor of Santa Cruz, and, quote, the father of the Santa Cruz Beach Boardwalk amusement park. The Swantons owned a lot of coastal land in that area. But Mrs. Swanton, who was collecting in the late 1890s and early 1900s, was kind of lost to time, like many of the women collectors in that early period. But I noticed the name of a collector, Bradley M. Davis, who collected many specimens in 1892 on the left there, and again in 1922. And I was curious how someone would collect 30 years apart with a gap in between. But unlike Mrs. Swanton, he was very easy to track down. He was a member of Stanford's first graduating class in undergrad in 1892. And then he left and went on to graduate school on the East Coast and eventually became a professor at the University of Michigan, a professor of botany. And as a professor, he made the trip back to Monterey Bay to collect some of the same species 30 years later. So it was easy to look him up. He was the president of the American Society of Naturalists, and he was well documented in the academic world. Professor Isabella Abbott was one woman collector who was well documented. She was a professor at Stanford at the Hopkins Marine Station, and her collections served as the heart of our time series during her tenure. She was collecting up and down the coast and literally wrote the textbook on Pacific Coast seaweeds, and this study wouldn't have been possible without her. So the objectives of this study were to, 1, demonstrate new value for underused herbaria that are widespread across many institutions. 2, generate records of the ecosystem from these seaweeds before instrument measurements were possible. 3, to establish repeatable methods for wide application that others can use to answer their own questions relevant to their systems. And 4, to encourage institutions to change herbaria operating procedures to monitor global change. This isn't a new idea for herbaria curators, but herbaria are often underfunded, so new collecting going forward has really declined in recent decades, and as well as funding for the preservation and collection maintenance. So our study was sort of in 2 phases. 1 was a methodology experiment followed by a historical case study. So for our phase 1, the experiment, before we could go into the historical collections, we had to first understand the effect of storage methods on the seaweed microchemistry. Is the tissue of this Macrocystis kelp here, pressed in 1922, comparable to a freshly pressed specimen today? Or did the chemical signatures change from the pressing and drying process? And then phase 2 was our historical case study. Once we knew how to interpret the historical pressings. And so we used these historical herbaria to reconstruct environmental time series prior to the start of instrument records. So for our phase 1 experiment, we collected seaweed in southern Monterey Bay at low tide. We collected 6 species to get representatives from each of the 3 major taxa categories, green, brown, and red algae, and also to represent morphological categories. So we had a branching red, a branching brown, bladed brown, etc. And we only selected species that were well represented in the historical herbarium. So when we moved on to phase 2, the case study, we would have a long and detailed time series. So you can picture our species. This is our bladed red algae, Cryptopleura, branching red algae, Gelidium, the pincushion, branching green algae, Chlodophora, bladed green algae, the sea lettuce, Ulva, our branching brown algae, a rockweed, Sylvetia, and a bladed brown algae, Macrocystis pyrifera, the giant kelp and the icon of the Monterey Bay Aquarium. We rinsed the seaweed in salt water and pressed the specimens in a traditional botanical herbarium press, shown on the left there. We also selected tissue from each species to immediately oven dry, shown on the right, instead of pressing, so that we could get near immediate results of the seaweed chemical signatures and then be able to compare to the process of pressing and drying. Once specimens were dry, we processed them in the Ocean Memory Lab and sent them to several laboratories for analysis. Of the pressed specimens, we repeatedly sampled the same organisms over time to look at the process of pressing and storage and see if the microchemistry components change or degrade over time. So we're sampling here at a few days after collection, 1 month, 2, 4, and 6 months after collection, and then 1 year after collection. And we pressed on these specimens on both modern and historical paper, archived paper, because historical manufacturing may have used acid or metals in that process. We ground the tissue and homogenized it with mortar and pestle and sent it to labs for amino acid analysis, heavy metal panels, and carbon, nitrogen, oxygen stable isotope analyses. So our results for amino acids, the top set of panels are macroalgae pressed on archived historical paper, and the bottom set were pressed on modern museum grade paper. And then each individual panel is one amino acid with data points for each species. So lowest curves are shown for all 6 species combined. The x-axis is days over 1 year that each species was repeatedly sampled, and the y-axis is in units of % weight. So the takeaway here is that first, for each amino acid, there's not a significant change over time in their value over that 1 year period. And second, that there's no difference between the historical archive paper on the top and the modern paper on the bottom for each of those amino acids. Similarly, for heavy metals, there was no difference between the archived and the modern paper. There was more variability within a species for some of the species and for some of the metals, such as lead or mercury. But there was not a large change over the length of the curing process from beginning to the 1 year end period. Similarly, for carbon, nitrogen, and oxygen stable isotopes, there was no difference between values on the archived paper on the left or the modern paper on the right. Carbon and nitrogen isotopes showed no change over 1 year of drying and curing, while oxygen showed an enrichment in the first couple months, likely due to humidity in our storage. So to summarize our phase 1 experiment results, did the historical paper alter the microchemistry of the pressed macroalgae? No. So we can interpret historical specimens without calibration. Did the pressed seaweed values change over the 1 year? Overall, no, but some species were more variable for certain metrics. These results from the experiment informed our selection of macroalgae species to use as an environmental proxy or ocean memory recorder for our phase 2. So for phase 2, the historical case study of 1 environmental record, we selected the Gelidium genus from the 6 that we used in the experiment, and nitrogen stable isotope ratios as our microchemistry metric. Gelidium was consistent for nitrogen stable isotope values, and gelidium was well represented in the herbarium. So we then dove back into the historical herbaria and sent samples off to the laboratory for nitrogen stable isotope analysis. We corrected our nitrogen stable isotope values for the season the gelidium specimens were collected in, and then related these values to several environmental variables before finding a strong correlation with upwelling using the Bacon upwelling index. And that is the figure on the left showing that relationship. When upwelling was high, the nitrogen stable isotope of gelidium collected in that time period was also high, indicating an increased growth rate. So the Bacon upwelling index is a measure of upwelling calculated from wind that began in 1946. It's the oldest and kind of coarsest version of these upwelling indices. And upwelling is the process of cold, nutrient-rich waters from the deep being pushed up to the surface near shore, and it happens seasonally. It's the dominant nutrient regime in the Monterey Bay, and the reason why the region is so productive. So we derived the relationship between upwelling index and nitrogen stable isotopes in gelidium using the specimens that were just from 1946 to present. We then hindcast the upwelling index values based on this relationship using the gelidium specimens from the herbaria that went back to 1878. So in this figure, the right half shows the smooth monthly upwelling index values from 1946 on using the wind recording instruments. The left half shows the hindcast upwelling values based on each little data point there is a gelidium specimen from the herbaria. So in mid-century, we see a noticeable decrease in the upwelling index estimate in the 1940s. And to look further into that, in this system, others, including researchers at MBARI, the Monterey Bay Aquarium's Research Institute, the partner institution of the Monterey Bay Aquarium, have documented regime cycles that alternate over multiple decades between forage fish species, the sardine regime during warm phases and the anchovy regime during cool phases. And these temperature phases are part of the Pacific decadal oscillation, a large basin-wide process. The decrease in upwelling that the seaweed documented correlates with the peak in sardine catch that happened in Monterey's famous sardine cannery era. And it's followed by a shift to an anchovy regime, which was the crash of that industry. So while the temperature regime is the dominant basin-wide influence, upwelling is a local feature that can impact forage fish as well. Strong upwelling favors large prey near shore that anchovies specialize on, while weak upwelling results in smaller prey that sardines specialize on. And the research supports this weaker upwelling pattern during a sardine regime, as seen here. So both the warm phase of the Pacific decadal oscillation, the temperature, and its associated weaker upwelling are predicted to lead to an increase in sardines, which is what Monterey experienced before its infamous crash. So in summary, for our case study, macroalgae revealed historical upwelling patterns that are predicted by these broader theories before instruments were available to document it. These environmental processes may have contributed to the sardine anchovy regime shifts. Overfishing was originally understood to be the cause of the mid-century sardine crash before researchers described these ocean basin-wide temperature oscillations. And then local upwelling as well, which our macroalgae documented. So in conclusion, this method can be used by other researchers to answer environmental questions using herbaria throughout the world and questions that are relevant to their systems that may be different from ours. Herbaria are underfunded and underutilized in conservation research. They're more frequently used in systematics. They're frequently used in teaching and identification. And there's some excellent work examining changing species distributions. But more questions can be asked, and continued collection is necessary to provide long-term records to allow researchers of the future to ask questions we cannot imagine today. Over 100 years ago, Mrs. Swanton or Bradley Davis couldn't have anticipated what we would be using their specimens for, and we similarly can't know how ours will be used in the future. This work would not be possible without the funding of the Monterey Bay Aquarium, the field and laboratory assistance of several people listed here, and the herbaria and their curators that supplied specimens would not be possible without their contributions, so we thank them. And thank you for taking the time to go through my talk.